I will be discussing DNA replication in prokaryotes. Here's a prokaryotic cell, and this is its circular DNA. The origin of replication on the circular DNA is rich in AT sequences and is recognized by proteins that can bind to the site and begin the replication process. When replication begins, two replication forks are made and move around the circle, eventually making a complete copy of the DNA. Gyrase and helicase play an important role in preparing the DNA for replication. Gyrase relieves supercoiling ahead of the replication fork, similar to someone with very curly hair getting a straight perm. Helicase is like the zipper that unzips the two strands of DNA. They work together like this to separate the DNA strands and prepare them for replication. Single-strand binding proteins, like toe separators while you're getting a pedicure, keep the strands separate by coating them near the replication fork. Next, RNA primase makes a primer out of RNA, complementary to the DNA. The primer is similar to a rope with several knots tied into it. The knots allow enzymes to easily grab on. DNA polymerase III adds nucleotides individually to a growing chain, like chain links being added one at a time. DNA polymerase III is a holoenzyme, which means it's made of enzymes combined with coenzymes. It has two core enzymes, a sliding clamp unit, and a clamp loading complex. So a primer is necessary because DNA polymerase III requires a 3' OH group to add a nucleotide. A primer provides the 3' OH so DNA polymerase can add nucleotides by forming a chain of phosphodiester bonds. The primer shown here is on the lagging strand, meaning that DNA is going to have to be synthesized in the opposite direction of the helix unwinding. The DNA polymerase III will latch on and synthesize nucleotides in the 5' to 3' direction. When more helix is unwoven, another RNA primer will be placed and the DNA polymerase III will attach nucleotides, stopping before it hits another primer, leaving a gap and creating many separate fragments. These are called Okazaki fragments. The other strand, called the leading strand, is simpler. A single primer can be placed and DNA polymerase III can continuously place nucleotides in the 5' to 3' direction, towards the area where helicase separates the strands. There are RNA chunks in the leading and lagging strands. How will this be fixed? This is the lagging strand as an example. It is the job of DNA polymerase I to remove RNA primers and fill in the gap with corresponding DNA. It recognizes the RNA chunks by its RNTPs, which are ribonucleoside triphosphates. After the primers are replaced with DNA, ligase enters the picture. Ligase is like a glue and can put broken pieces back together. Ligase creates a phosphodiester bond between the 3' OH of one Okazaki fragment and the 5' phosphate end of an adjacent Okazaki fragment. If DNA polymerase I could no longer perform its exonuclease function, RNA primers would be left attached to the DNA strand and any mistakes in DNA synthesis would not be repaired. RNA primers being left in the DNA would prevent transcription or replication of the strand in the future.